No, carry on. You've still got 2 minutes, don't worry. Okay, welcome back, everybody. I hope you managed to get something nice for lunch. So moving on to session number 5, where we're focusing on pest management. So this session is a session of 2 halves, really. We've got 3 speakers in this session, and we're looking at 2 topics. So cabbage stem flea beetle and then BYDV. So we're joined by Sasha White from ADAS today, who'll be talking to us about the AHDB-funded research projects. And then we've got 2 of our AHDB-sponsored PhD students, Claire and Maria, and they'll be sharing with us the latest results from their projects. So to kick things off, I'd like to introduce to the stage Dr. Sasha White, who is a senior research entomologist at ADAS. Thank you, Sasha.
Afternoon, everybody. So yes, I'm going to be presenting some research on cabbage stem flea beetle that we've been doing over the last year. It's a 3-year project. We're about just started the 2nd year of it. It's called Reducing the Impact of Cabbage Stem Flea Beetle. It's a piece of work that's funded by AHDB, but we also have funding from a wide consortium of industry organisations, and they're all listed there. The work is led by ADAS, but also in partnership with Harper Adams University and Cotton Farm Consultancy. Now, the project has got 2 main aims. The first is to get a better understanding of the biology of the pest and its phenology. So that's the timings of its life cycle throughout the year. And within that, there's a whole lot of sub-objectives going on in it. But essentially what we want to try and do is understand how we can better predict when things are going to happen and how severe the problem will be with a pest. The second main aim is to identify effective integrated pest management approaches and test those on farm. And for today's talk, I'm going to focus on that second aim. So because of pyrethroid resistance, we now don't have great control from insecticides with cabbage stem flea beetle. Resistance is really widespread in the country, certainly across England, and it's very, very common. So we aren't able to rely on control from these insecticides. So we need to really start thinking more and more about non-chemical control approaches. And listed here are all of the non-chemical control approaches. There may be a couple that I haven't thought of, but here are the main ones. And they're categorized by whether they help avoid the pest. So something like a trap crop will draw flea beetles away from your cash crop and into the trap crop. So that's an avoidance strategy. Other ones will help mitigate against the damage. So for instance, if you choose a variety that's got good autumn vigor, that might help the crop grow away quickly from adult feeding damage and therefore mitigate against the damage. So as part of the last AHDB funded project, which finished a couple of years ago now, we allocated each of these a traffic light. And that was after kind of reviewing all of those and the research that was out there on all of them. Where we had a green traffic light, that meant that approach provided pretty reliable control and there was good evidence for it. There might be some need for some more research to fine tune it. Where the light was amber, it meant that the work was showing, the research had shown that we could maybe get moderate levels of control, but we needed more research to try and optimize that and understand it better. And there, where there was a red light, it meant that control was really unproven and we needed to do significant amount of research to really bring it up to a point at which we can get the most out of it. By the end of this project, we wanted to get all of these approaches to those traffic lights on the right. So we want to really get them to the point at which we know where they work best and where we can get some reliable control from them. So I'm just going to focus on 3 trials that we've been doing so far in this project. And I should emphasize that there's a huge number of trials that are happening. Last year alone, in year 1, we had 19 trials that occurred through ADAS and also through the project partners. This year, there's at least a dozen. So I'm going to focus on 3 of them. And the first one is the effect of sow date. So in the previous project, we did some modeling work of a very large survey data set to try and pick out which factors best determine flea beetle risk. And one of the most important, and certainly the most important thing that growers could change, was sow date. And I'm just going to take you through quickly what we found in that work. So on the left, what we found was that moisture was very important. So making sure that you drill into the right conditions was critical. Regardless of what sow date you're aiming for, it's really, really important to try and respond to the conditions. So what we found was crops that were drilled and then followed by a lot of rain suffered significantly less damage than crops that were drilled in dry conditions or only had a small amount of rain afterward. But sow date also has a direct effect on the susceptibility to the flea beetle, and that's illustrated with that graph on the right there. So what the modelling found was that the crop stage at the point at which flea beetle migration occurs has a very important effect on its vulnerability to damage. So what we found here was that where a crop was at or before emergence, when flea beetle migration started, then they tended to get much higher levels of damage than crops that were at the cotyledon stage, or even less damage if the crops were at a later stage. And really what this is the basis of this big shift that we've seen in recent years to much earlier drilling, because essentially what you're doing is you're drilling the crop, it's emerging, and it's well established by the time that flea beetle migration kicks in, which is usually towards the end of August or the first week or so of September. But it isn't quite as clear as that, unfortunately, because early sown crops tend to have higher levels of larvae in the autumn and the spring than later sown crops. And this is illustrated with this graph here. So this is the larvae per plant in the autumn. And we can see that crops sown in the first half of August have much higher larval loads than crops sown in September. And certainly in the second half of September, you've got a fraction of the number of larvae in these crops. So this is the autumn. It's a very similar trend in the spring. Now, the big question mark about all of this is based on survey data and modelling work, and there's always a level of uncertainty there. So we wanted to carry out a trial to really see whether what we see in that trial is what we saw in that modelling work. So last year we drilled 3 adjacent fields with different drill dates. So we had a 26th of August drill date, a 6th of September, and a 15th of September drill date. This was over in the West Midlands. And each of those fields had 2 varieties drilled into them. They're both Bassef varieties. It's InVigor 1035 and InVigor 1140. And basically those 2 varieties differed in their autumn and spring vigor. Well, what we found immediately was the 6th of August drill date was lost to flea beetle. It was wiped out pretty much immediately. So the host farmer then re-drilled that field on the 1st of October. And that was also subsequently lost to flea beetle. I'm just going to put these drill dates into context of the flea beetle migration. So what we can see here is the green line shows the cumulative flea beetles caught in traps over time in relation to those 4 drill dates. And it's worth saying that these traps weren't actually in these fields, they were nearby, but I think it's still pretty indicative of what was happening. So what you can see, certainly with the 6th of September drill date, that crop would have emerged and coincided with that peak of flea beetle migration coming in the 15th of September. So it would have been at a very vulnerable stage when that flea beetle migration started, which probably explains why it was lost. And then the 1st of October drill date, which was drilled into that same field that was wiped out, was also lost, probably because of all the flea beetles that were already there waiting around, having eaten the previous crop. So we were left with two fields left, the 26th of August and the 15th of September. So what do we see in these fields? Well, firstly, we can see the effect of the sow date and variety on adult damage over on the left here. What we can see, so the blue bars are the InVigor 1035, the green bars are InVigor 1140. And there were some small differences between the varieties, but certainly the very, very clear difference was that the crop drilled on the 15th September suffered significantly less damage, 2 to 3 times less damage than that crop sown a field away 3 weeks earlier. And essentially what we think is happening here is that this crop was emerging as flea beetle migration was tailing off, and most of those flea beetles had moved into earlier drilled crops. In terms of larval load, it's a similar story. So this is the larvae per plant in March. And again, we can see some small differences between the varieties, but a very, very clear difference between the drill dates. So a 4 to 5-fold reduction in larvae by sowing 3 weeks later. So what this trial shows is that we've got some really good supporting evidence for that survey work and the modelling work. And it's clear that sow date is critical for determining flea beetle pressure. What we know is that there's a high risk window for drilling oilseed rape, which is roughly from the 3rd of August all the way through to the middle of September, which gives us 2 low risk windows, before that and after that. And actually we can see that, you know, there is a trend, as I mentioned earlier, towards earlier drilling. So this is on the right here, a graph which shows the proportion of crops sown over different periods in the last 3 years. And this is a study, it's the DeKalb study by Balear. It involved interviewing 150 or so farmers over which included tens of thousands of hectares of oilseed rape. What we can see is a big shift. The blue bars are this year, and we can see a big shift towards earlier sowing. I would say that sow date is so important that really we need to use sow date as the framework for the other management approaches that we use. So if you're sowing early, then your main risk is from larvae, because these crops will tend to have higher levels of larvae, so you should be focusing your work on managing the larval pressure. If you're sowing later, then the adult damage is a greater risk, so control measures should focus on minimising adult flea beetle damage. So I'm just going to cover a couple of other trials that we've looked at, and the first of which is companion crops. So there's been a lot of talk about companion crops recently, and so we've set up a line trial this summer where we had 3 treatments. So the first treatment was winter oilseed rape on its own, and then the next 3 treatments are with different companion crop mixes. So the first one is with buckwheat, then we have buckwheat and berseem clover, and then we have buckwheat, berseem clover and fenugreek. And really what we wanted to see was, do we get a greater benefit by having more companion crops, having a mix of companion crops in there. All the companion crops were drilled on the 15th of August, 5 days before the winter oilseed rape. And we think that's important to get the companion crops up and away, so they're providing that protection to the emerging oilseed rape crop. So what did we find? So in terms of plants per metre squared, this is the graph over on the left, we can see that we've got 2 growth stages, growth stage 14 and 15, and really there were very few differences, and they certainly weren't significant between any of the treatments. So the companion crops weren't hindering establishment or providing a benefit, certainly in this regard. But then when we look at adult flea beetle numbers, we did see an effect. So here we can see this is the total flea beetle counts caught in water traps in each of the areas, and we can see we have significantly more flea beetles caught where there's an oilseed rape monoculture, and less where we've got the companion crops, and it was a significant reduction where we had buckwheat on its own. And then when we looked at the percent leaf area lost to flea beetles, also we saw some significant impacts on damage. So highest levels of damage where we've got an oilseed rape monoculture, and then significant reductions in damage under each of the companion crop mixes, with the lowest damage in the buckwheat and berseem clover mix. So what this shows is really that companion crops are seeming to provide some benefit. So we're seeing fewer flea beetles where there's a companion crop mix there. There's less damage as well. And broadly speaking, these results are similar to a trial that we ran in the first year of the project, which was last year. But it is worth emphasising that both these sites had pretty low flea beetle pressure overall. So the question is, do we still see a similar level of reduction in flea beetle damage where the pressure is higher? And it's also worth saying we will be looking at larvae and indeed yield in this trial as well. Okay, so the last project last trial I just wanted to touch on here was a trial looking at organic amendments. So this is a trial we set up this summer. It's another line trial as well. Here's the layout. And in this trial, we looked at 3 different treatments. So we had the control, which was inorganic nitrogen applied to the seedbed. And then we had the second treatment was digestate applied to the seedbed. And then the third treatment was a delayed application of the digestate, which was to the establishing crop, and it was timed to coincide with the migration of the flea beetles. So what did we find in this? Well, here we can see the effect on the plant stand on the left. Here we've got the plants per metre squared at growth stage 12 and 13 in each of the 3 treatments. And we did see a significant effect here. So what we found was that where we had seedbed digestate, we had significantly more plants per metre squared than where we had inorganic nitrogen at the seedbed. In terms of adult flea beetle numbers, the trend wasn't very strong, but there was a slight trend for lower levels of adult flea beetles in the area where we had applied digestate to coincide with migration of the flea beetles, and it was significantly reduced, lower levels compared to certainly the seedbed digestate on one of the assessment occasions. And then in terms of percent leaf area lost, we also saw some significant effects here. So what we can see is that damage was significantly lower where digestate had been applied to the seedbed, certainly compared to the seedbed inorganic nitrogen treatment. And we also saw significant reductions in damage where migration or digestate was applied to coincide with migration compared to the first assessment in the seedbed organic nitrogen treatment. So here we can see that seedbed digestate is improving establishment. We think this is because of the available, readily available nitrogen, and digestate generally is reducing damage and flea beetle numbers. But it's worth saying these aren't huge reductions. So they might help a bit, but they probably won't solve the whole problem, or at least not in all situations. What we think is happening, certainly in terms of flea beetle activity, is that it might be masking the crop from migrating flea beetles because they're attracted to the smell of the oilseed rape, or they might be actively deterred by the digestate. Okay, so there's a lot of other work going on in the projects. In terms of the trial work this year alone, we've got trials looking at stubble length, varieties, cultivation methods, seed rates. But outside of the trial work, we're doing things like trying to identify the factors that determine flea beetle migration. We're investigating the environmental conditions that affect egg hatch and larval development. We're also going to be looking to better understand the impact that larvae have on crop development. And very lastly, I just want to let you know there's also a sister project going on here. This is the Nyab-led project, which is also started last summer. And essentially what they're doing, amongst other things, is supporting growers to run their own trials and then helping them collect that data. And what that will allow us to do really is test these approaches on as wide a scale as we can, really. And I think that kind of goes back to what Neil Paley was mentioning in that first talk this morning about having the opportunity and the need to test IPM strategies on a wider scale as we can. So just to summarize, the project is looking to try and identify the role of IPM, try and help us understand where these non-chemical approaches work best, where they don't work as well, how we can optimize them. Sow date is critical to flea beetle pressure. We can see in these trials that companion crops and organic amendments are showing some promise. This is good because we're actually seeing non-chemical control approaches increasingly being adopted by industry. And this is another graph from that DeKalb survey, which is showing the percentage of crops, oilseed rape crops, that are using some of these non-chemical control approaches specifically for flea beetle control. And we can see a good adoption of a lot of them, really. And sow date would be right up there as well if that had been an answer, I'm sure. And I think we are getting better at managing the challenge for flea beetle. You know, I think the damage has been low for the last 2 years. I think weather has helped, but I think as an industry, we're getting better at managing this pest. And again, this is reflected by another graph in this same DeKalb survey. And we can see here what we've got is the proportion of crops experiencing different levels of flea beetle pressure over the last 3 years. And we can see this year, 57% of crops were reported as having little or no challenge. So yeah, thank you for listening, and just to thank all the funders and the project partners and all the people that have been involved in this work.
Brilliant. Thank you very much, Sasha. I think you said towards the end there about the importance of trying things out in different parts of the country. And what I really like about that project is the focus around on-farm trialling and larger scale trialling as well. I don't know if it came through, but part of that project was around having ring-fenced funding, and the work had to be completed on some of our strategic farms. We've had a really nice link between our AHDB funded research projects and then our knowledge exchange programs as well. And that leads us nicely on to our next presentation from Claire Horu from Harper Adams University. She's a PhD student at Harper, sponsored and supported by AHDB Cereals and Oilseeds. And Claire's looking at another and what we could consider a new tool in the toolbox for managing cabbage stem flea beetle, and that's focusing on biopesticides. Claire's in her second year and has most recently been working with one of our former monitor farmers, Adrian Joint, who's at Bridge North. So I'll hand over to you, Claire. Thank you.
Hi, everyone. Thank you, Emily, for introducing me. So yeah, I'm going to talk about my work on biopesticides to control the cabbage stem flea beetle in oilseed rape crop. So I'm going to go relatively quickly on this part because Sasha already talked a bit about this. But yeah, the cabbage stem flea beetle is a major pest in oilseed rape crops. Both the adult and larval stage can damage the crop. The adult feeding on the leaves and creating damage called short hulling, and the larvae mining the petioles and in their later stage moving into the main stem, and they can potentially lead to crop failure. They were originally controlled by neonicotinoid insecticides, but they've been banned by the European Union in 2013. So the control of cabbage stem flea beetle has been reliant on pyrethroid insecticides. But as Sasha said, cabbage stem flea beetle are now widely resistant to pyrethroids. And those products are also known to be harmful to non-target organisms. So there's a real need to identify alternatives to conventional insecticides. And the focus on my project is to look at biopesticides with the objective to include it in an IPM program in the future, hopefully. So I've done several laboratory bioassays using Antoine pathogens, so bacteria, nematodes, and fungi. I've also looked at different physically acting products, and I've also compared these data with a conventional synthetic insecticide. So I've chosen a pyrethroid for that. And I've also selected the best option I had from the lab and tried them in the field. I'm just finishing this trial, so it's very recent. And I've looked at leaf damage and at the adult cabbage stem flea beetle density in the field as well. So the first bioassay I'm going to present is with the entomopathogenic bacteria Bacillus thuringiensis tenebrionis, which is known to affect different species of Coleoptera. So I've tried 3 different formulations, 2 from one of my funder, Certis, and another one from Andermatt UK. So these are still in development, that's why they're coded. And I've also compared these with a control with water. So I did this by dipping oilseed rape leaves in bacterial solution, then giving them to my beetles to feed in the lab. And then I've recorded cumulative mortality. So we can see here on this graph the mean number of cabbage stem flea beetle dead after 12 days, depending on the formulations. And we can see then it doesn't seem to be any significant difference between the control and between the different formulations. And I didn't get more than 50% mortality overall. Then I tried antipathogenic nematodes, so 4 different species provided by BASF, which I thank for these formulations. So the nematodes were diluted in water and brought down to 3 different concentrations, so 4,000, 10,000, and 40,000 nematodes per milliliter. And then 1 milliliter of each solution was dropped into a petri dish and then exposed to Cabbage flea beetles adults. And again, I've recorded the cumulative mortality every 2 days until 8 days post-inoculation. So this is a very busy graph, so I'm going to start with 1 species at a time. So we can see that the control was relatively low, with no more than 25% mortality compared to the first species here, Heterorabies bacteriophora, which was very effective. For example, we can see that even at the lowest dose, after a couple of days, it's yielded more than 50% mortality. And when using the highest dose, it yielded 100% mortality after just 2 days. Then we have the other species. Steinema capocapsae was also quite efficient, but a bit slower to act. We can see that after 2 days, we didn't reach more than 30% mortality, regardless of the dose, but still reached quite high mortality after 8 days, even at the lowest dose, with almost 75% mortality. Then Steinema feltiae worked quite well at the highest dose, with 80% mortality after only 2 days, but was less efficient at the lowest dose without more than 50% mortality. And then Steinema macrosae was identified as the least effective species, with no more than 60% mortality and quite a lot of variations in between the replicates. So in between all these species, I've decided to select Heterorabies bacteriophora and Steinema feltiae to try in the field. So we're going to see that in a couple minutes. Then another bioassay I've done is with the entomopathogenic fungus Overia bassiana strain GHA as a commercial formulation, botanical WP, again provided by my funder Certis, that I've applied at 3 different rates: half the field rate, and double the field rate, with again control with water. And the fungal solutions were directly sprayed on the beetles, and then I recorded cumulative mortality until 14 days. So we can see that there was no dead in the water control, so this is quite good, and significant differences between the control and all formulations. But there's definitely a better effect with double the field rate, with more than 50% mortality, and slightly lower efficacy with half the field rate and field rate, but still more efficient than water. Then going away from pathogens, I've tried 2 different physically acting products. The first one is Flea, which is a fatty acids-based product and developed by Alpha Biocontrol and Balear, which I thank for providing this product. And another physically product, I can't disclose the content because it's still in development, and again provided by Certis. So again, 3 different concentration: half the field rate, and double the field rate, and the control. And in addition, I've examined the cuticle of Cabbage flea beetle with a scanning electron microscope to see if we could see the effect of these products on the cuticle. And again, I've sprayed the product directly on the beetles and recorded mortality. So we can see the results here. Still, again, absolutely no dead in the control with water. Flea was particularly efficient. For example, we can see that after 1 day at the field dose, it's more than 80% mortality. And when double the field dose was applied, it was 100% mortality after just 1 day. And the Certis formulation was a little bit less efficient, but still more efficient than the control. And at double the field dose, we still reached more than 60% mortality. So both these products I've tested in the field. And here we can see pictures taken with a scanning electron microscope. We have here the control with beetles only sprayed with water, and this one is the cuticle after flipper being applied. So we can see that the integrity of the cuticle seemed to have been disrupted. So it seems to have a visible effect, which is quite nice. And then I've tried the conventional insecticide Hallmark Xeon, which is a lambda thialytrine provided by Syngenta, and again applied a higher field rate, and double the field rate, and sprayed directly on the beetles. So I had not exactly the results I was expecting, because Cabbage beetle is supposed to be resistant to this, but yeah, the field rate I had 100% mortality. But after thinking about this, we realized that spraying the product directly on the beetle was not at all reflective of what would happen in the field, and we've decided to repeat this experiment in the future by using a vial coating bioassay. So coating vials, leaving it to evaporate, and then allowing the beetle to walk on the vial and then record mortality. So I'm going to repeat this bioassay in the future with a different method. Now, for the field experiments, quite a lot of treatments, but mostly what I've been tested in the lab. So flipper, the other physically acting products, the conventional insecticides applied 3 times and only 1 time as it should be, but I also applied it 3 times as the other treatments to see if there was a difference. Then Botanigard azatine, which is a botanical biopesticides, which I've tried in the lab without success, but it's been shown in other studies that it could have an impact when it's applied with another Antoine pathogen. So I've applied it with Botanigard. Then I've also, of course, had a water and untreated treatments to compare, and 2 nematode treatments. So the antimopathogens and physically acting products were applied 3 times once a week, and for the nematodes, it was applied 4 times at 4 different rates. And this trial was done at Apley Eastad Farm in Shropshire. So I've assessed the initial leaf damage before treatment using this guide over there. And I've also assessed the leaf damage after the treatment to see if there would be a difference. And something I'm not presenting here because still in the process of doing it is collecting and dissecting the plants to count the number of larvae per plant. So this is something I'm looking forward to see the results of, but I can't really say anything about this yet. And I've also looked at adult cabbage stem flea beetle density with yellow water traps placed in the field. So just a quick photo here, we can see a couple of cabbage stem flea beetle in the trap. So we definitely found some. So this is the initial leaf damage. So before the treatments on the 22nd, 24th, and 29th of September. So the plants were still quite young with cotyledons and first true leaf stages. And we can see that it goes slowly from 10% to around 15%. So there's definitely an increase in time. And here is from T1 to T10, so no nematodes. No nematodes are a separate trial. So this is 7 days after the first treatment. We can see that the plants had grown a bit, and there doesn't seem to be any differences between the treatments and also compared to the water treatment and the untreated treatments. It looks like the Hallmark treatment seems to be a little lower, but I'm not 100% sure because the stats are not completed for this. So this is what I get from the graph, but I'm not 100% sure. And when we look at 20 days after the first treatment, so the plants have grown quite a bit as well. Still, doesn't seem to be too much differences between the treatments. This is T7 is Flipper plus Hallmark. Seems to be lower, so there seems to be a good combining Flipper with a conventional insecticide seems to have an impact compared to Hallmark itself, by itself. And when you apply Hallmark only once compared to 3, there seems to be a better effect as well. So there's definitely no use of applying pyrethroids repeatedly. But yeah, compared to the other picture, we can see that the box plot seems to be a bit narrower, so there seems to be less variation between the blocks and a more even trial. These are the nematodes, so Steinheima feltiae and Heteroditis bacteriophora at 3 different doses. We can see that there doesn't seem to be any differences between the doses and in between the different species, and again, around 20% mortality, which is similar to what we had here. So it's 6 eggs after the first treatment, and this is 19 days after the first treatment. So again, still around 20%, and doesn't seem to be any differences between the different species. And this is quickly the number of adult Cabbage and Flea beetle per water trap throughout the trial, so which was almost 2 months. And we can see that it was very variable, but we never had more than 14 beetles per trap, which is much lower than the threshold for treatment, which is 100 beetles per trap. So overall, this crop was doing quite well in terms of pest pressure. That's why we have relatively low leaf damage. So in summary, I had encouraging results in the lab with 2 species of entomopathogenic nematodes, Bovea bassina and as well Flipper. However, these bioassays with lambda cellotrine definitely need to be repeated. And even though there seems to be no apparent significant effect of all this treatment in the field, the methods need to be improved, especially the time of spraying, because we know that most of the biopesticides I've been using are living organisms such as nematodes and fungi, and they definitely prefer to be sprayed without any UV radiations. So spraying in the evening, that was the goal originally, but we realized that logistically it was complicated. So we didn't do it, but that's definitely going to be done next year, hopefully. So yeah, next I need to finish dissecting those plants to see larval density. I'm also going to test new formulations of the product in the lab and look at their effect on CSV critical, like for Flipper. And at the end, before the end of my project, by November 2022, I will of course look at cost benefits of those biopesticides to see if they can economically if it's feasible to actually apply them in the field by farmers, and also possibly effect on non-target organisms. And yeah, hopefully before I finish my PhD, I'm hoping to do another field experiments with an improved method. So quickly, I'd like to thank you for your attention, and my sponsors as well, AHDB, Certis, and the Agri-Food Charities Partnership, my supervisors, Tom Pope and Heather Campbell, and also my collaborators at the University of Warwick, Dave Chandler and Jill Prince. Thank you.
That's brilliant. Thank you, Claire. And it's really fascinating to see that progression of your work from the lab work that you've been doing into the field experiment. So I congratulate you on that, because I know it's not easy. So we're getting our money's worth out of Sasha today. We're going to leave Cabbage and Flea beetle behind, and Sasha's going to talk us through management of aphid and BYDV risk in winter cereals. Thank you, Sasha.
So yes, this is another project that's been funded by AHDB. It's again a 3-year project. We're into our final year now, actually. I should say that we've also been funded by Syngenta, BASF, KWS, and Limagrain as part of this work. And it's a project that's led by ADAS, but we are also collaborating with Martin Williamson at Rothamsted as well. So this project has 2 overarching aims. The first is to develop effective and economic monitoring methods for BYDV vectors. And the second is to develop decision support tools to help growers and advisors in making management decisions for barley yellow dwarf virus. In today's talk, I'm going to firstly talk about the autumn pressure from BYDV vectors that we've had so far this year, and that's going to touch a little bit on some of the bits of work that we've been doing in that first aim. And then for the second half, I'm going to describe a decision support model that we've developed as part of the second aim. So what's the pressure been like this year? Well, this is data from the Rothamsted insect suction trap network. Here we've got count data, or catch data indeed, for bird cherry oat aphid or Repellosyphon pedi. Bird cherry oat aphid is one of the two main vectors of BYDV. The other one is Tibio navini or grain aphid. So these are the catch data for two of the suction traps. So on the left, we've got Rittle, which is in Essex, and on the right, we've got the Newcastle suction trap. What these show is the numbers of aphids being caught at these two suction traps throughout the year. The green bars show the 10-year average, and the white line shows last year's catch data, and the red line shows this year's catch data. Now, what I want to draw your attention to is really this September onwards period here for both of these sites. What we can see is that this year, more aphids are being caught in these suction traps, more Repellosyphon pedi were being caught in these suction traps than last year. And although these differences might look huge, because this is on the log scale, these differences are, in terms of real numbers, very, very, very big. And also it's worth saying that this is a trend that by and large we've seen across the country, across the suction traps across the country. So certainly there's more aphids being caught in suction traps this year than last year. What's been happening in crops? Well, as part of the project, every year we monitor crops across the country for the presence of the two aphid species. So in this year, we had 12 crops that we monitored in Yorkshire, 12 in the east, 12 in Devon, and 10 in the West Midlands. And in each of these crops, we went in 4 times in the autumn, and we counted aphids on 100 plants. And what you can see here is the totals that we found in each of these areas, and also you can see the breakdown of the species. So RPs, Repellosyphon pedi, and SA, Tibio navini. So in Yorkshire, we found a total of 37 aphids in all of these crops. In the east, 72 aphids. In the West Midlands, 79 aphids, and in the southwest, just 5 aphids. It's not a huge return, really, on quite a lot of monitoring of crops. I mean, essentially, we found 1 aphid for every 95 plants that we checked. So that seems to be quite low pressure, seemingly. But I'm not sure that's the whole story, because what we also did in these crops is we put out water traps. So in each crop, we put out water traps on 2 occasions. And on each occasion, we had 2 sticky traps and 2 yellow water traps. And here we can see very different numbers. So we've got the totals here. So 150 aphids were found in those water traps in Yorkshire, 589 in the east, 340 in the West Midlands, and 56 in the southwest, which is an average of 3 aphids per trap, although obviously some traps had a lot more aphids than others. I think what this indicates really is that putting out traps in fields is a really good way of monitoring your pressure, because whilst you can go and look at aphids on plants, because of the way that aphids migrate into crops, they migrate into the crops and they will infest a very, very, very small number of plants in those crops. So it's very easy, just by pure chance, to miss the plants that have the aphids on them, and then therefore get a underestimate of your pressure in your field. So it certainly looked like in-field trapping was a really good way of determining the pressure. So we looked at water traps and sticky traps, where either of them are better than the other. Well, here on the right is we've got the total number of aphids caught in each of these 2 traps this year. And you can see we caught 2 to 3 times more in the water traps than we did in the sticky traps. And that's a trend that really we've seen for the last 2 years of the project as well. That it does look like water traps are a better way of monitoring these BYDV vectors. So how about some year-on-year comparisons at these sites? So on the left, you can see the on-plant counts. This is the total number of aphids we counted in all of those sites over the last 3 years. In 2019, we found very, very few aphids. Last year, we found a lot more, and this year we found slightly less than last year. But on the right, we've got the data from the in-field traps. So this is the total number of aphids we caught in these water and sticky traps for the last 3 years. And we can see for the last 2 years, very few aphids have been caught. And this year, we've caught a huge, relatively huge number. And I would say this is the more reliable indication of the pressure in field. So it certainly looks like this year we've had more aphids in suction traps and also more aphids in field as well. But it's not all bad news, because it looks like that the percentage of aphids carrying the virus is lower this year than last year. So as part of the project, Rothamsted, so Martin Williamson at Rothamsted, has been testing aphids caught in suction traps to see whether they're carrying BYDV. And this graph shows the total percentage at 5 suction traps where this was carried out over the last 2 years. And what we can see is at all of the sites, a smaller proportion of aphids are carrying the virus this year than last year. And it's also interesting that we can see some trends between the sites as well. So for instance, over in Hereford and at Star Cross in Devon, we tend to get higher percentages of aphids carrying the virus than at other sites, certainly Brooms Barn, for instance, which is in the east of the country. What we think is happening here is that in places like Hereford and the southwest, there's a higher frequency of permanent grassland, and this acts as a reservoir for the virus and indeed the aphids to then take those into winter cereals in the autumn. Okay, so that's the sort of the seasonal update and of aphid pressure and BYDV pressure. I'm just going to move on now to a model that we've developed as part of the project. So this is a decision support tool which really helps growers understand and advisors understand when sprays are needed, and also what the benefits might be of different drill dates and also using tolerant varieties. And it's based on 2 previously developed models that were developed in the 80s and 90s, and we've essentially updated them and merged them together. And we've called it the ADAS Crop BYDV Assessment Tool, or ACROBAT for short. Now I'm just going to take you through the input page for the model. It's very ugly at the moment because this is a version that we use as researchers. If it was to become more widely available, then we would obviously make it much more user-friendly. But this is a really convenient way of showing what goes into the model. So in the top left, we have input data on aphids and weather. So the aphid data can come from suction traps or from data that you've collected from a specific field. We also need to let the model know what crop it is. Is it wheat or barley? That's important because barley tends to be more severely affected by BYDV. The model needs to know the percentage of aphids carrying the virus. And at the bottom left here, we've got some crop agronomy information. So what is the cost of the treatment? What's the expected yield? And what's the expected grain price? And this is all important because basically the model makes a cost-benefit calculation of whether a spray is of any need or not. At the top, we need to let the model know when the crop was drilled and what the plant population is. And then we also need to know the average winter temperature for the previous year. And that might seem a bit odd, but the reason we have this is because this is a good indicator of the proportion of bird cherry oat aphids that are migrating into cereals in the winter. So just as an aside, bird cherry oat aphid has essentially 2 life cycles. So in the winter or in the autumn, a proportion of the population will migrate to bird cherry trees where they complete the sexual phase of their life cycle. In fact, that's the majority of bird cherry oat aphids do that. A smaller proportion will keep their life cycle continuing in cereals, and it's those individuals that will migrate into winter cereals in the autumn, and they continue an asexual life cycle. And the previous winter temperature is a good indicator of that. At the bottom, we've got some information about the field location, so how close is it to the sea, and what is the surrounding land use. And these are important because they're good indicators of BYDV risk. And once we have that information, we run the model. And the model outputs various things, but the salient bits of information are the proportion of crop that's infected with the virus and whether a spray is recommended or not. And I'm just going to take you through some example outputs of the model and some scenarios about what can change BYDV pressure. So here we've got a graph that shows the proportion of plants in a crop infected over time. This is a wheat crop that's drilled on the 1st of September, so inherently high risk. And what we can see is we've got 5 different scenarios. So what these scenarios are the percentage of aphids that are carrying the virus when they migrate into the crop. So the red line shows if we had 50% of aphids that were moving into the crop, 50% of those were carrying the virus. What we see is a very big, very rapid build-up in the proportion of plants being infected over time. And in this case, a spray was being recommended by around the 19th of September, which is about a week after the crop emerged. So a very high-risk situation here. As the proportion of aphids carrying the virus reduces, so the rate at which the plants get infected slows, and the spray recommendations are delayed. So that by the time we have just 1% of aphids carrying the virus, the spray recommendation is delayed by about a month. Okay, so here we've got another output, but this time is looking at the effect of temperature. So it's the same layout and essentially the same crop, but we've got the red line is a baseline temperature, the green line is 10% warmer, and the blue line is 10% cooler. And in this situation, we can see that in the baseline situation and the slightly warmer condition, a spray has been recommended at about the same time, around the 6th of October. Where the conditions are cooled, the spray is recommended is delayed by about a week or so. And essentially the reason for this is because those cooler conditions are slowing down the reproduction of the aphids and the rate at which they develop in the crop, and therefore the rate at which they move out into the crop and therefore spread the virus. And then as a final scenario, here we've got the same layout, but this time we're looking at the effective aphid pressure, the numbers of aphids. So R-Paidae, that's Rhopalocyphon paidae or Burcheriot aphid. And so here the blue line is a baseline number of R-Paidae, and then all the other lines are reductions in R-Paidae numbers. And what we can see here is with the baseline, we're getting a spray around the 20th of October. But then as we reduce the numbers of R-Paidae migrating into the crop, so the rate at which plants are infected is slowed, and indeed so the spray recommendations are delayed, so that by the time you've got 95% less R-Paidae migrating into the crop, by the time we got to the end of October, no spray had been recommended. And I would guess, from our understanding of the model, a spray probably would have been recommended somewhere in late November, maybe early December. So you can sort of see what the model's doing. It's essentially helping us get an idea of when a spray might be needed in the autumn. And whilst we develop this model, what we need to do really is test it. We need to validate it. Is it telling us accurate predictions of what's happening? And we're validating the model in 3 ways. So the first way is we're surveying untreated crops to see whether the model correctly predicts the amount of BYDV we see in those crops. Secondly, we're running tramline trials. So these are naturally infested tramline trials where we have 3 different treatments. Some tramlines get sprayed based on recommendations from the new Acrobat model. Other tramlines are sprayed based on recommendations from the TSA model, which is already available through AHDB's website and other sources. And then there's other tramlines which are untreated. So we had 5 of those trials last year, but because these were naturally infested trials, BYDV pressure was very low last year, these trials showed us really very little. So this year we've taken a slightly different approach. We're still running 4 of these trials, but we're also doing an inoculated plot trial, which is this third option at the bottom. So this year we have an inoculated plot trial where we're inoculating plots with, each plot with 600 Burcheriot aphids, and these aphids carry the path strain of BYDV. In this trial, we've got susceptible and tolerant varieties, and overlaid on top of that, we've got 4 treatments. So we've got sprays as recommended by the Acrobat model, sprays as recommended by TSUM, calendar sprays, and then an untreated control. And that, by the way, on your right is an example layout of the tramline trials. Okay, so here's some pictures from that inoculator trial. And on the left there, you can see the trial layout. Those are all the plots. All the white points on those plots are where we inoculated the trial with the aphids, and that's an inoculation point at the top. We gave them a little bit of shelter for a week or so to protect them from the rain. And then at the bottom there, you can see all those lovely aphids have moved over onto our crop plants, which was exactly what we wanted to happen. And indeed, on the right here, you can see we've got BYDV symptoms already. Now, it's too early to present the data, but what I can tell you is across the 5 trials this year, the T-sum model has recommended 11 sprays and the Acrobat model has recommended 3 sprays, which sounds good as long as it still provides good protection. So that's what we're going to be looking at next in this work. So just to summarize, this year's aphid pressure is the highest that we've had since the loss of neonic seed treatments, but the proportion of aphids carrying the virus is lower, certainly than last year. In terms of monitoring, it does look like putting traps out into your field, and water traps are an effective way of monitoring for BYDV vectors. We've developed a model, the Acrobat model, that can help guide chemical and non-chemical BYDV management decisions. And this model is currently recommending less sprays than the T-sum model, but we do and are validating it in a variety of ways. So just to thank you for listening, and thanks to our funders, our project partners, and all these people that have had a very big role in the project.
Thank you again, Sasha. So Sasha has mentioned in his presentation spray recommendations in terms of timings of sprays. And our next presentation is going to look at whether we can target those sprays in terms of where we put them in the field, specifically using trap cropping. So I'm really pleased to welcome to the stage Maria Leandro, an AHDB Cereals and Oilseeds funded PhD student at Harper Adams University. Thank you, Maria.
Can you hear me? Fine. Okay. Thank you. Hi, everyone. So I'm Maria, and I'm a first-year PhD student at Harper Adams University, and I'm gonna be talking about my project, what are the aims of my project, and at what stage we are right now. And as Emily said, we're gonna be looking into improving crop monitoring and use of trap cropping as novel approaches to the IPM of BYDV vectors in winter cereals. Just a short introduction. I'm not going to repeat myself too much, as Sasha already was talking about BYDV before. But Balear Dwarf virus is a widespread, economically damaging luteal virus that affects a range of crops, and including cereal crops. And the symptoms are chlorosis, stunt plant growth, and also reduced stress tolerance, which translate as yield losses. And the main vectors in the UK are the grain aphid and the Balear aphid. And the aphids will usually migrate in autumn. That's when crops are most vulnerable to BYDV. And the virus strain that's more prevalent in the UK is BYDV PAV. There's 7 different strains, but I'm mainly looking at this one. And AHDB estimates that losses in untreated crops would average 8%, but they can be as high as 60%. And without effective treatment, BYDV could cost the industry around £140 million in wheat alone. And just to summarize today, what are the challenges behind BYDV management? First of all, as you all are aware, there's a decreased insecticide availability as we no longer have neonicotinoid seed treatment. This was the main treatment used to prevent BYDV. And also, there is insecticide resistance to pyrethroids in the grain aphid, and tolerance has also been reported in the Balear aphid. And also, there's a lack of competitive, tolerant, resistant germplants. There is one resistant variety, Wolverine, and there is one tolerant variety, but that's about it. And also another challenge is obviously climate change. We're gonna be expecting more unpredictable weather, and if this is gonna benefit the vectors or even benefit virus spread or not, we are very unsure. So we do have to be ready to act in whichever scenario we're gonna be expecting the next years. And obviously, you know what integrated pest management is. So it's just a reminder that it's a sustainable, science-based decision-making process. And it will involve different tools, but I want to highlight here that these tools are aiming to detect, manage, and mitigate pest risk, and while minimizing economic, health, and environmental risks. And my project is looking into that bit of the pyramid, which is monitoring, forecasting, and warning systems. And my research questions at the moment, at least, is if we could exploit the use of varieties that are more attractive to cereal aphids as trap crops within an IPM program to improve vectors management. And if we could improve in-field monitoring of BioIDV vectors through the use of aphid-specific semiochemicals. So just a short framework of my project, we're looking into basically these two aspects of IPM, prevention and detection. And prevention, we are looking into trap crops. As I said, we're gonna be exploring wheat varieties that are believed to be more attractive to aphids as trap crops. I'm gonna go more deeper into it in the next slides. And also combining trap crops with conservation biocontrol. And in order to look into the detection, we also want to exploit plant volatiles to improve trap crops for early detection of BioIDV vectors. As you know, once the virus is in the field, it's very hard to do something about it. You can just wait and try to do something in the next season. So if we can improve early detection, we can take the actions before the damage is done. So just to show a little bit what I've been doing for this year, I've been looking into, first of all, BioIDV diagnostics, as I need to carry out my trials in the lab and in the glasshouse in the field. I need to be sure that I'm using good diagnostics, BioIDV diagnostics, both in aphids and in the crop. And we've been using ELISA, but it's a very time-consuming, it's expensive approach. So we're moving into diagnosing BioIDV using PCR. And I've also been doing a landing behavior experiment, which is, I'm going to show you the results in the glasshouse. And we're also starting this season, we are doing field trials to evaluate the use of old wheat varieties such as Maris Huntsman as trap crops, and if there is any difference in aphid abundance in different wheat varieties in the field. So my first experiment is a landing behavior experiment. That's basically the objective is to investigate the potential of wheat varieties to be used as trap crops, and we are looking into which variety could be a preferred host for the BioIDV vectors. And we're using highly attractive varieties such as Maris Huntsman. It is a variety that some of you might know. It was widely grown in the UK in the 70s and 80s when severe aphid outbreaks were recorded. So there is not much in the literature. It's more farmers' knowledge that this variety, which is not a semi-dwarf variety, it's an old variety, it would attract more aphids because there was this pattern of more outbreaks. And then when breeding program moved and now we have semi-dwarf varieties, the aphid outbreaks are way less severe than they used to be. So we want to see if we could potentially use some of these older varieties as trap crops. And we also include in the resistant variety, Wolverine, to see whether there is any preference in both species for any particular variety. And all the other varieties are AHDB recommended list varieties. In total, I was looking at 16 different varieties. And I'm doing this essay because someone else, Dan, who already did a similar experiment, and it's not published, his work, but he found some interesting results with the different colors. So the blue ones are modern semi-dwarf varieties that at the time he did the experiment, they were all in the recommended list from AHDB. And the yellow ones are heritage winter wheat. And then the green ones are old varieties that were grown, as I said, in the 70s and 80s. And he found that there was a striking difference between the number of aphids landing in these older and heritage varieties as in comparison to modern semi-dwarf varieties for the grain aphid. And that's basically what I did, similar, but I did it for both vectors, both the bird chariot aphid, firstly here. And well, unfortunately for this species, I didn't find any significant difference yet. And the aphids I use for this experiment, they are BYDV free, but I also want to try with BYDV infected aphids to see if they would show any behavioral difference into landing in different varieties. We do see there was a tendency after 4 days that both the old varieties that I included were Flanders and Maris Huntsman. They were among the most attractive, if I may say, but overall there was not very high numbers of aphids of these species landing in general. So for this species, I can't say anything for now. But for the grain aphid, we had a bit more interesting results. There was difference between different varieties. All the dark gray ones, by the way, they are AHDB recommended list varieties. And then the light gray are Huntsman and Flanders. And here I'm showing the results 24 hours after I released the aphids, and then 4 days. And we can see that overall Flanders was more attractive than Huntsman. And in this case was more attractive than all the others. And then 4 days later, there was a bit less, the number was a bit lower, but could still include in most attractive. And unfortunately, I didn't see anything with Maris Huntsman as Dan saw, but I'm still carrying out this essay. And as I said, I've used virus-free aphids, and we're not sure what was the virus status of the aphids that Dan used. So I want to explore this with also BYDV carrying aphids to see if they're going to show any difference in behavior and any preference towards semi-dwarf varieties, any variety in particular. What is interesting, though, is that the variety overall, there is a group of varieties that for both species, they were the most attractive. There was a higher number of aphids landing in those varieties. And Wolverine included, which is the BYDV resistant one, was also, we could see a higher number of aphids as compared to all the AHDB list varieties like Barrow, Crusoe, Skyfall. And so this here, after having a look at my data, I decided to have a look at the different number by pod position. This was a fully randomized experiment, so every time I would repeat it, I would change the position of the varieties within my tents randomly, and I could see that some pod positions for both varieties, but for both species, but because the birch ariote aphid, there was not a lot of numbers, then we can't see anything significant. But for the grey aphid, we can see significance. And I could observe that there was a group of pod positions between 10, 11, 12 that always had higher numbers. And in the end, we conclude that probably there was a lot of light influence. And we want to repeat this experiment in a better setup where light will not gonna be of any influence. And so yeah, that's basically it for my landing experiments right now, but I'm still gonna repeat that. I'm gonna test the infected aphids, and hopefully we can see something interesting happening. And well, it's very nice to see that in the glass house and to test things in the lab, but obviously we want to see what happens in the field. Sometimes we do things in a more controlled environment, and then when we go to the field, the results are completely different. So I'm also prioritizing doing field trials since the beginning of my project. So I'm only in the first year, but we're already doing field trials, testing trap crops, and I've got 3 different sites at Harper Adams University where we are testing Maris huntsman. That's one of the old varieties as a trap crop. And the way we're doing, we have a commercial field with Zayat, which is a AHDB recommended list variety. And we are testing both the same variety as in the field, in this case Zayat, as a trap crop. So is a plot planted drilled in the edge of the field. And we are testing Maris huntsman, and I'm monitoring this with yellow sticky traps, both in the field, in the main field, and in the trap crop. And basically I'm looking to compare if we're gonna see different numbers of aphids in the edge as a whole, so we can validate that trap crop, it will be effective. And also if there is a difference in between using a trap crop that is just the same variety as you use in the field, or if really using a old variety that we think it could be more attractive to the vectors, we're gonna find different results. So I started monitoring this since we drilled, and I'm gonna carry on this monitoring until April. So that's the threshold for the disease after the crop is well established. BYDV is not such a big problem, so we want to monitor, keep an eye as soon as we drill in the earlier stages of the crop. And another experiment I'm doing in the field, I've just did some plots with three different varieties, and I'm also monitoring them using sticky traps. And I just want to see if there is any difference in aphid abundance in a AHDB recommended variety, semi-dwarf variety, Wolverine, that showed higher numbers landing in the glass house, and Huntsman, which is an old variety. I deliberately chose varieties that they have different parental lineages. So also this experiment is ongoing until April, and hopefully we can see some interesting results later. And well, I still have quite a way to go with my project. So just to show you what I'm planning on doing, I want to investigate the olfactory basis of host plant selection by aphids. This is basically, I want to look into behaviorally active plant volatiles that could be related to these wheat varieties that maybe are more attractive for aphids. If there is any volatile or a particular blend of volatile that could be used to improve sensitivity of trap catches in the field. Because today we just use either sticky traps or water traps. They're not specific for aphids, or there's nothing, it's not there's no any chemicals used on that. So if we could have something that we would be sure that is catching more aphids directly to these vectors we're looking at, we could improve this early monitoring. And secondly, as I said, I'm carrying on a few tests looking into the trap crops, but I also want to combine that with flowering strips and monitoring. And I also want to record the natural enemy response and what is the potential of combining trap crops with conservation biological control. And finally, I'm looking to investigate when or how this apparent preference for landing on old varieties, such as Huntsman, was lost during breeding programs when we had this shift towards semi-dwarf varieties. And that's it. I just want to thank all my supervisors and my sponsors and my partners. And thank you all for listening. I'm happy to take questions.
Brilliant. Thank you, Maria. If I can invite Sasha and Claire to join Maria on the stage, and we'll get the questions loaded up. We'll have a quick fire question rounds. Thank you. So question for you, Claire, first. Is there a danger that the nematodes could kill beneficials as well?
Nematodes are relatively specific, but they could kill beneficials, but the goal would be to apply them when the beetle are active and hopefully when outside of the window of activity of beneficials such as parasitoids. So yeah, there will definitely be, the target will be to apply them when beneficials are not active, and hopefully I'll be able to try this in the future, yeah.
Brilliant, thank you. Question for you, Sasha, I think. Is there any research into how many larvae per plant oilseed rape can tolerate and produce a viable yield?
This is something we've looked at quite a lot, and I think the answer is it's variable. So we've seen some crops that could tolerate very few larvae and have a yield impact, and then some crops that can have much higher larval loads and still get good yields. It's one of the things we want to try and find out a bit more in the project. I suspect that bigger plants are better able to tolerate more larvae. And we've done some work with previous field labs, innovative farmer field labs, where we looked actually at defoliating crops in the winter to control larvae. And there was one farm over in Norfolk where the defoliation, it reduced larval loads, but it didn't really have a great impact on his yield. And in fact, he yielded very well over 2 years, actually. And his plants had 40, 50 plus larvae per plant in them in the spring. And what he was doing is he was drilling at a very, very low seed rate so that these plants in the spring were bushes, essentially. And so there's a possibility that just larger plants are better able to tolerate it. But we just need to drill down into it and try and get some controlled experiments running to better understand it.
Brilliant, thanks. For you then, Maria, you mentioned in your presentation about monitoring and the importance of monitoring and that the traps might not have been designed specifically for monitoring aphids. We've got a question here. In-field aphid trapping, if you had to choose one for growers to use, would that be the sticky traps or the water traps? Any thoughts on that?
So, yeah, Sasha might be able to help me. As he said, according to his results, water traps were more effective. In my case, because it's a very intense monitoring, I have quite a lot of plots and I'm doing intensive monitoring to really see the difference everywhere. I have as much data as I can. So for me, sticky traps are better because I can just keep them in the freezer and looking at them later. Mm-hmm. And it's absolutely fine. But for water traps, you need to kind of assess them as you go, basically. And it might be better for farmers to look at water traps I would say. But yeah, I think it really depends of the logistics. I don't know if you want to add.
I mean, I think we're hit the nail on the head there. Yeah, I mean, I think it depends on what you're trying to achieve. If you're going to monitor it there and then, maybe water traps are better.
Yeah.
Water traps have their issues. They can get knocked over. They can, if there's a lot of rain, they can flood and and that sort of thing. So, but certainly, we seem to be finding more aphids in water traps than sticky traps.
Yeah, because I'm going to compare the numbers I have in one place, both with the sticky traps. I still can draw some conclusion, even if perhaps if I have water traps, I would have more aphids. But if I have more aphids in one situation than in another, with the trap crop or without the trap crop, I'm using sticky trap for both. So for me, it works well, and I can keep it in the freezer. Can look at it at a later date.
I guess at the end of the day, it's better for us to be doing something rather than nothing. So don't get too hung up on which one we do, as long as we're out there looking in the field. Right, you can see we've got some more questions, but I'm conscious that we promised you an opportunity to grab a cup of tea at this point in the day. So I think our speakers will be with us for the rest of the day. So you've got 6 minutes, if that's okay. Trying to keep it to time. I don't want you to be away at the end of the day any later than we'd promised. So if you want to grab a quick cup of tea, and then we'll start again at 15:55. But just join me in thanking our speakers. Thank you.